Okay. So hello everybody, thank you for being today with us from a webinar organized by the African Pharmacogenetics Network. This will a part of the series of the Pharmacogenetic Network webinars. I hope you will continue with us and keep following up with us for the next webinars too. So today we will have an interesting session about the pharmacogenetics of sickle cell disease therapeutics in Africa with Dr. Mnika. She is a lecturer in the Division of Human Genetics at the University of Cape Town. Her research interests are primarily centered around the pharmacogenomics of sickle cell disease, therapeutics, and the genetics etiology of inherited kidney disease in Africa. So, Dr. Mnika, please go on. The floor is yours.
Thank you so much. I just want to apologize in advance because the office that I'm sitting in, it's close to a nitrogen tank. I always apologize for this because there are people that are working next to this building, so you might hear some noise, but I hope it doesn't get worse.
No problem. Okay.
Yeah. I'll just start sharing my slide. I won't see anything when I share my slide, so I think we can save the question for the last, or you can type as you think of any question, and then we have a chat at the end. Okay, cool. Let me do that. Please confirm if you can see my screen.
Yes, it's clear.
Thank you so much. Thank you for the invitation. The talk that I'm going to share today, actually, it's my PhD work. Currently, I have about, I think, 4 years. I finished this work, but I think it's very important for me to share this work because I feel like it's one of the novel work that we've done in pharmacogenomics, specifically for sickle cell disease. And this talk, I'm going to share about genes, or maybe let me say variants that are associated with pain and drug metabolism in sickle cell patient, and also look at the hydroxyurea post-transcriptional gene expression. I'm just going to start with an introduction just to say the global distribution of sickle cell disease, it's more prevalent in Africa. I think everyone knows this. And you can see other parts of the country are shaded. It's not that it's original from those parts of the country. This was because of the slave trade in that happened long time ago, and recent migration. People are moving around, as you know. We seek for a job, we move around. So, but initially, this disease is more prevalent in Africa. And I just want to explain what happened when you are sickle. So I think this is a basic genetics. I think everyone knows, but I think it's important to do the house rule so that we are on the same page. So sickle cell disease is a monogenic mutation. That means it's a change on it's a single change whereby your glutamic acid is replaced by valine. And then when this amino acid is replaced, you can see that on this, I'm not sure whether you can see my mouse pointer, laser.
Yes, it's clear.
Okay, I just want to do the laser so that, okay, I'm not sure whether you can see. So you can see the hemoglobin S, the one that have hemoglobin S that are sort of, they have mutation. You can see that it forms a long chain. This is because of the change of the charge of the amino acid. So it tends to cluster together and form this long chain. And this long chain of the hemoglobin, it affects the structure of your red blood cell, moving from your round shape to a concave shape. Most people, they call it sickle cell, but then I think in most part of the Africa, sickle cell term, it's not well used. I think recently the sickle cell disease ontology, they had a new term, which is banana cell, which makes sense for everyone in Africa to say banana cell because they will know what you're talking about. So these banana cell, they tend to get stuck in your blood veins. And when they get stuck to the blood veins, there's a lot of pressure to move the blood through. And this results in like your vaso-occlusion crisis or organ damage. And this leads to other clinical phenotype. And sickle cell disease is known as the multisystemic disease. What does it mean is that it affects a lot of system in your body. That means there are a lot of clinical phenotypes that are coming from different systems that will result by you having a mutation in the hemoglobin gene. So these are the part of some of the clinical phenotype that are associated with sickle cell disease. But however, in Africa, the most one, it's your stroke, and the other one will be your kidney dysfunction. Most of our patients, they have stroke, they have kidney dysfunction. Some of them, they'll have leg ulcers. Because sickle cell disease is a multi-system disorder, it's very complex. By not looking at the sickle cell disease and other clinical phenotype, but also looking at genetic modifiers. So we have the genetic modifiers, we have the non-genetic modifiers. I think the well-known genetic modifier for sickle cell disease is alpha thalassemia, so which is co-inherited with sickle cell disease, when you have compound inheritance whereby you have your alpha thalassemia and sickle cell disease. Your severity differs with the person that has only sickle cell disease. We can see that if you have two deletion, you are less severe in terms of phenotype, but then the more deletion you have, the more severe your phenotype it is. And then there are haplotype that have been characterized, and you can see that it ranged from Arabia Indian haplotype to Bantu haplotype. Bantu haplotype is a severe haplotype, which is mostly prevalent in the Central Africa. And then we have your fetal hemoglobin, which is a well-known genetic modifier for sickle cell disease, because patients with sickle cell disease, they have low level of fetal hemoglobin. So you can see if you have low level of fetal hemoglobin, you have less phenotype. And then if your fetal hemoglobin, if your fetal hemoglobin is low, then you have severe phenotype compared to the high level of fetal hemoglobin. There are some other modifiers which are environmental that also contributes towards the severity of the sickle cell disease. But I just want to highlight only one, which is the infections. So most of these patients, because their immune system is very weak, they're prone to more infections compared to us people who don't have sickle cell disease. I just want to highlight the therapeutics that I use for sickle cell disease, because this disease is mostly in Africa. So African patients, mostly they use African herbs and plants because they don't have access to the standard therapeutic approach. And then we have those standard therapeutic approach, which is hydroxyurea. Hydroxyurea has been known as a golden standard for sickle cell disease, but there are other type that are used to manage sickle cell disease. I think one of them that is not here is Vesilito, which is a new medication that was approved, I think, in 2019. So there's also a cutting edge technology. I think everyone know CRISPR-Cas9, which is a gene editing tool that has been introduced, I think, since 2017 for sickle cell disease, to make sure that they see if they can cure sickle cell disease. However, in Africa, all these cutting edge tool, it's very limited to us. Not to say that hydroxyurea, which is a medication that was approved long time ago, is still very limited. I mean, the availability is very limited in our African patient. I just want to share guidelines because I'll be talking about pharmacogenomics. I just want to share guidelines that have been published on the use of hydroxyurea. You can see the red block that I've highlighted here. They don't have a personalized dosage or personalized medicine. For sickle cell patient, they use a universal scale, regardless whether you have this gene that is like you are a poor metabolizer or you're a fast metabolizer. There's just like a universal, which is like a traditional way of giving medication, one size fit all. But then I want you to pay attention to the availability in low resources, which is the last column. I think it says it's limited availability. In Africa, even though we have high burden of sickle cell disease, but then the sickle cell management in Africa is still poor. There's also other medication that these patients take. Remember, I mentioned that they are prone to infection. So they also take penicillin. The most one that is taken by sickle cell patient in Africa, it's malaria prophylaxis, which is also available, but in other parts of the country it's very limited, specifically in Africa. I just want to share what has been published recently, I think by Kevin. So Kevin published this data looking at the frequency of sickle cell disease compared to the number or to the countries that have sickle cell disease treatment. If you look at this, I think it's A, you see that the ones that are shaded, it's where the treatment center are available. And then when you look at B, you see that there's a stem cell transplant center. So the only centers that are open for the stem transplant, it's Nigeria and South Africa. Mind you that South Africa has low number of sickle cell disease. Actually, most of those patients, they're not original from South Africa. They're coming from other parts of Africa. So Tanzania, they recently launched the stem cell transplant. Stem cell transplant, it's an approved technology that cure sickle cell disease, but however, it's used in a limited setting because of risk that are associated with, and also it's difficult to get donors. We've studied sickle cell disease a lot, but I just want to show you this map which illustrates nicely. You can see that the first case was reported in 1910, but then you see that hydroxyurea was approved 1998. The whole decade of sickle cell disease, there were no other medication that were approved. And then we see in 2017 that other medication came in place and then they were approved in 2019. But when you look at this map, you see limited numbers of pharmacogenomic study. The only studies that were done for pharmacogenomics is when they were doing clinical trial, but none that was replicated in African population where there's like mostly severe uh, um, sickle cell disease, like burden of sickle cell disease. Why are we worried about pharmacogenomics in sickle cell patients that are using hydroxyurea or any other medication? If you look at this, I think everyone in this meeting knows this picture that currently in our setting, we are using the traditional way of giving patient medication, which is one size fit all, regardless of whether you are poor or you are a good metabolizer. However, the goal of pharmacogenomics for us is to make sure that we move away from this one size fit all and move towards the precision medicine. I think that's one of the things that sort of prompted us or motivated us to do studies on pharmacogenomics of sickle cell disease therapeutic, because we saw that there's limited data, specifically in Africa, on pharmacogenomics of sickle cell disease therapeutic. One day we want to see this whereby there's a genotyping where before they give you medication so that they can give you the dose that is specific for you, or they give you an alternative drug which is specific for you. So that's our long-term goal. Hopefully we'll get there. I just want to explain how hydroxyurea works, because I'm going to focus more on hydroxyurea because it's the golden standard. It is mostly used in Africa compared to the other medication. But when I look at the literature, I saw that the other medication that were recently approved, there's limited, actually there's no studies that have tried to understand the pharmacogenomics of those medication in African population. So for hydroxyurea, you can see the first block, it's whereby there's without hydroxyurea, and then the second block, it's with hydroxyurea. You can see that the production of the red cell with a minimal level of fetal hemoglobin, there are a lot in this block, and then there's few that have high level of fetal hemoglobin. So why fetal hemoglobin? Because fetal hemoglobin, it changes when you grow older. Sorry, I think I changed by mistake. It changes. So the level of fetal hemoglobin is high as while you're still a baby. As you grow older, after 5 years old, it drops. But then when you have sickle cell disease, that's where you see more clinical phenotype or manifestation of the sickle cell disease because of that level of fetal hemoglobin. And then the the hydroxyurea, it works in a way that it compensates the level of fetal hemoglobin. So when you see in this block 2, you see that hydroxyurea, it kills some of the cycle of the cell. So that means there will be less, or there will be fewer red blood cell with the minimum level of fetal then increase of the one that has high level of fetal hemoglobin. So as I've mentioned before in that first, I think in that first second slide where I talk about pathophysiology, you see that these red sickle cell, they block and then they result in these other clinical manifestation. But when you have hydroxyurea, your red blood cell, they're not 100%, but however, there is some compensation of that fetal hemoglobin level. That means you improve in terms of the clinical manifestation. There are some of the side effects that have been published that are associated with the use of hydroxyurea, specifically in sickle cell patient. You can see the short-term one as the decrease of your blood count. So these ones, it's a trial and error, like we said before, that it's a one-size-fits-all. So they give you the standard dose and see how you respond, and they'll keep on adjusting your dosage based on the improvement of your blood count. And then there's some that are long-term. I think the one that is also a problem for a sickle cell patient is the decrease of the sperm count. So most of them, they are very conscious about like using hydroxyurea because of that. I think we know that in Africa, we love big families. I think that's why they sort of get to be hesitant to use hydroxyurea, because it has been reported that it does decrease the sperm count. However, it has never been evaluated in African population that it really does affect that. And also there's another one, which is the productive. So it has been said that people that have sickle cell disease, when they are pregnant, they can't use hydroxyurea because it can lead to early miscarriage and all that. And most patients, they just stop using hydroxyurea because of that. But then when they stop using hydroxyurea, that's where they have these severe clinical manifestation or crisis that can affect the baby either way. But this has not been studied well enough in Africa. It has not been proven whether this is an issue. Hence, we need to do a lot of pharmacogenomics in sickle cell disease. So why pharmacogenomics in sickle cell disease therapeutic? I think we all know that we want to do the pharmacogenomics because we want to identify the genetic variants that sort of are different among patients that influence the treatment response. And also it's important for us to look at these three main areas where we look at efficacy, the safety, and the optimum dose. We also want to look at the cytochrome P450 enzymes, which are responsible for drug metabolism. This is not yet well studied in hydroxyurea, specifically for sickle cell disease. And remember, I mentioned that our patient, they are prone to infection. So that means they use other medication beside hydroxyurea. That means we still need to sort of tap into drug-drug interaction, which is the area that is not yet well characterized when it comes to hydroxyurea in sickle cell patient, to see how these patients are responding to this medication. What can we adjust? What are the enzymes that are responsible for this drug metabolism? I've put this picture on this, and I think this it's coming from a literature review. We don't have, actually, let me not say we don't have. We have limited number of studies that have done experimental work in terms of pharmacogenomics in sickle cell disease, specifically looking at hydroxyurea. Most of the studies that are published, that are out there, it's a review, which means the study that review what has been done, and most of those studies are not done in our African settings, are done outside Africa. So we saw this, and then we sort of thought that it's also important for us to contribute towards the pharmacogenetics specifically, because we all know these other genes that are responsible for the fetal hemoglobin production. For example, your BCA11, your MIB, your KFL1, they're all responsible in the production of fetal hemoglobin. However, the mechanism of all this is not well known. We don't know whether there are other pharmacogenetics genes that are sort of play a role in the production of the fetal hemoglobin, or either in the drug metabolism of this hydroxyurea. And another paper also sort of combined data from different papers, whereby they look at the baseline of fetal hemoglobin. What are the genes that have been known for the baseline of fetal hemoglobin? And then what are the genes that are associated with drug metabolism of hydroxyurea? To be honest with you, limited numbers are coming from Africa with these studies on pharmacogenomics. And then they've also come up with sort of like a hypothesis that those ones that are rapid metabolizers, they'll have high baseline of fetal hemoglobin, and those ones that are poor metabolizer, they'll have low baseline of fetal hemoglobin. Now we know that fetal hemoglobin plays a role, but however, the mechanism is not yet well clear. How does it play a role? Then we thought we will contribute towards the community of sickle cell disease by looking at the genetics that predict the painful vaso-occlusion crisis in patients that were coming from Cameroon. Remember, in that map that I showed you, I told you that Cameroon doesn't have any sickle cell disease treatment site or initiative that is happening there. We thought we'll look at that to see what are the genes or what are the variants that are associated with painful vaso-occlusion crisis. The reason why we look at this VOC, I think it's because we all know that sickle cell patient, the hallmark clinical phenotype is pain. So everyone that you meet along the road who is sickle cell patient will tell you that pain is the most clinical phenotype they experience. And we also thought that it would be nice to look at the post-transcriptional signatures to see how is it triggered when you patients are treated with hydroxyurea, particularly looking at the microRNA expression. So I'm going to share these two key aims with you, what we have done. I think for our aim, we thought to achieve our aim, we needed to first look at the literature to see what has been done, looking at globally in Africa, and also narrowing it down to Cameroon, which where patients were coming from for my study, and also look at these genetic variation that are associated with pain and in Cameroon. And the last part was sort of to look at whether we look at ex vivo or in vivo, to look at the microRNA expression under hydroxyurea, how does it act? What genes are they linked to it? Do we have any microRNA that are targeting genes that are responsible either for gene expression that will sort of help us to understand, or maybe to get other biomarkers for therapeutic in our sickle cell patients, specifically in Africa, because other parts of the country, they've done some of the study, but however, the burden is in Africa, and then there's limited number of studies that are done in our patients. So we thought these objectives will sort of help us to achieve our aim. So what we started with the first one that I've mentioned, to do the literature search. I think when we are doing the literature review, we wanted to see what has been done in pharmacogenomics, specifically for sickle cell disease, not only for hydroxyurea, but for other therapeutics that are used for sickle cell disease. And then I think we came up with the title that says "Not Yet Ready for Global Precision Medicine," because we saw that there's limited data, regardless where you are in a country, in pharmacogenomics for sickle cell disease. Our key terms that we used are the one that are highlighted red. What we did, we used a strategy whereby we went to the MESH database, and we look at the synonyms of these key terms, and then we created search term that we use in different databases to search papers. We did manage to get data from the papers that we search, and then we try to sort of separate them into different groups. So the first one was to look at the variants that influence the pain in sickle cell disease. We did see a lot of studies that were coming out on sickle cell disease and pain and vaso-occlusion crisis. But however, these studies were not from African settings. And we also, I'm sorry about that noise, and we also see that there are other studies that has done gene variation that are associated with morphine. Morphine is given to patients with sickle cell disease when they have these vaso-occlusion crisis, or they have crisis. When they get to the emergency unit, they give them morphine to sort of tone down the pain, because as I've mentioned that pain is the main key clinical phenotype for a patient. So we saw a number of studies that were looking at morphine metabolism, but there were no studies that look at morphine metabolism in sickle cell patients specifically. We also wanted to see if there is any study that has done or that has looked at the genetic variation that are associated with hydroxyurea induced, because we know that hydroxyurea, it increased the level of fetal hemoglobin. It has been seen that if you have a high level of fetal hemoglobin, you are responding well to the medication. That means you have less clinical manifestation of phenotype. We saw that there's a lot of studies that have been done across for the fetal hemoglobin production. And most of the genes that are in this table is the genes that we have already investigated in our research group in sickle cell disease as well. And it was nice to see that genes that we see in our patient, or variants or SNPs that are associated with fetal hemoglobin production, we see them that have been reported to be a key genes that we have to look at when we're looking at fetal hemoglobin production under hydroxyurea. But what was our take-home message from this whole entire search of papers? We saw that indeed there is limited number of studies that look at pharmacogenomics of sickle cell disease therapeutic, regardless where you are. But when you look at Africa, we saw that there's a very limited numbers of studies that focus on sickle cell disease in terms of pharmacogenomics. And we saw few available studies, and those studies were large studies. However, they were not replicated in other population, because we know that genetic diversity in Africa, it's very high. So a patient that is coming from Congo might not necessarily need the same dose with the person that is coming from, maybe, let me say, Tunisia. So we have a lot of genetic diversity. I think we also highlighted the key views. I think because my supervisor or my PI for the project was very clever. When I look at these key views, I see now everything that we've indicated in that paper. Exactly, it's what is happening now in a sickle cell field. So what we suggested, we said we are anticipated to see an increased number of the global burden of sickle cell disease. Not that it's people that have sickle cell disease that are being born more. It's because now there's more surveillance or newborn screening that has been done in lower countries and Western countries, and the use of hydroxyurea is increasing compared to previous years. When I look at the literature, I see now there are a lot of countries within Africa that now are looking at newborn screening in terms of to see how can they manage the patient that have sickle cell disease at early stage. And then we also saw that there will be more studies that will look at pharmacogenomics of sickle cell disease therapeutic. Indeed, after we published this paper, there were a lot of papers that were looking at pharmacogenomics of sickle cell disease, and also looking at how can we work towards the personalized medicine. There are a lot of papers, it's just that I choose few. This one is a recent paper, the one that it says towards genomic medicine. It's a recent paper, but it seems like the field has not been progressing that much after we've done our paper on pharmacogenomics of sickle cell disease. We can see that there's still lack of data that is from Africa, whereby they're looking at, you can say, genome data, whereby we're trying to find our novel SNP that are associated or specific to African populations, so that we are able to sort of filter down those genes that are associated or that are responsible for drug metabolism in sickle cell disease therapeutic, specifically in Africa. So I've been calling Africa all the time because I'm an advocate for Africa. So after this, we thought it would be nice to look at those genes that we have reported in our literature review, and just to see if we do screen those genes and do like genetic characterization, what can we see in our population? We then did a study. This study was stemmed from the literature review. All those genes that we have identified, or all those genetic SNP that we have identified that are associated either with pain or vaso-occlusion crisis or fetal hemoglobin level, were included, and we did like a genetic characterization just to see if are we seeing this SNP in our population. But before we did that, we went to the 1,000 genome, and we wanted to see how prevalent are they in our population, so that we don't waste time and look at the SNP that are not prevalent. I think someone who's into genomics will tell me that 1,000 genome, it doesn't represent Africa. That's a story for another day. But however, as African, we need to work towards building African genome. And I think the SAMRC, they're working towards and building the genome that we'll be able to use as African scientist as a reference. So the reason why I just want to go back and highlight that we did the literature review, because I think in my research group, you don't do any scientific work, or let me say experimental work, without you looking what has been done, what are the gaps, how can you fill in the gaps. I think that's how that's the culture that we grew up in, that you have to look at the literature review so that you are able to identify the gaps, so that you can fill those gaps that you said they are the gaps that you see. So for us to fill the gaps that we've identified in our literature search, we did a study whereby we're looking at the genetics and the clinical predictors of painful vaso-occlusion crisis in sickle cell patient. So these patients were from Cameroon. So what we did a targeted genotyping. So most of these SNP were genotyped using TechBen assay. Some of them were outsourced using a mass array. Then we validated this genotype using Sanger sequencing, just to make sure whatever that we call homozygous, it is a homozygous. Whatever we call heterozygous, it is a heterozygous. So we did that. When we did our analysis, we saw that there was a correlation for number of vaso-occlusion crisis with gender or with sex. So we saw that there was a correlation, and it has been reported in papers that gender does influence pain. Someone will say maybe men are more stronger, or they don't express pain, and then women, they express more pain. It's I think it's a discussion for another day. And then we also saw that there was sort of a correlation when you look at the hemoglobin level and number of vaso-occlusion crisis, which has already been published on literature, that the number of hemoglobin, or the level of hemoglobin in your blood count, it does all correlate with number of VOC, whereby if you have more, or you have high level of hemoglobin, you'll have less number of VOC compared to the one with low number of hemoglobin level. And then we also saw that there was a correlation with the BMI, with the vaso-occlusion crisis. I guess this one, maybe someone can argue and say maybe it's the high cholesterol in people that are obese, which is still need to be investigated, because we didn't see any study that has sort of reported this, the significant or the association with the BMI with number of VOC. My apologies, I think I've cut these figures. You don't see the VOC on the y-axis. My apologies for that. And then we saw the white blood cell. White blood cell have been reported that is associated with number of VOC in the literature. So what we have replicated here is something that has been published out there. And then for number of hospitalization, remember the plan was to look at the genetics and the clinical predictors. We also look at the hospitalization to see, are there any correlation with hospitalization with number of VOC and consultation as well. We saw that there was a correlation. But I just want to take you back that this data that we've collected on the number of hospitalization and the number of VOC, number of hospitalization was taken from the clinical record, but however, the number of VOC was self-reported, which we'll talk about at the end of this at the end of this objective, just to indicate the number of limitation of the study that we had due to the self-reported of VOC. So when we look at the genetics, after looking at the clinical predictors, we look at the genetic predictors. We included all the genes and the SNP that were sort of associated with pain or vaso-occlusion in our literature review that were reported. And we saw that there were some few that were associated with number of vaso-occlusion crisis, and we saw that there were some that were associated with consultation and hospitalization. So we also had the borderlines. I think someone will ask, why have you bolded this one, which is 0.08? So we bolded this one because we thought it's very important for us to note the ones that are at borderline, because this might not I mean, looking at the sample size that we have for our study, so we didn't want to let it out. We wanted someone who's reading this paper to be able to include this SNP when they're doing replication study, so that we can sort of narrow it down and take it out and say this, it's still not significant in other population within Africa. So we look at the known genetic modifiers that have been sort of published. So we did replicate some of the genetic modifier that have been reported to be associated with number of VOC. You can see alpha thalassemia, as I've shown you in that first slide, that the more deletion you have is associated with the severity of your disease. And we also saw that the upper L1, there was an association. I think people that are into kidney disease, they'll know that this is a risk allele for people to develop kidney disease either now or later stage. So we thought it's good for us to know this genetic modifier, so that we can also put people that are at risk to develop kidney disease, even if though they don't have at that stage. So it's also nice to use it as a proxy to say, if you have the G1, G2, your management needs to be looked closely, because you might develop kidney dysfunction at a later stage as a sickle cell patient. We also look at other genes that are responsible for fetal hemoglobin production. Actually, what we have produced here, it replicates what has been reported on the literature, which is not new. However, it was good that to see that whatever that was done on the literature, it has been replicated in our study. As I've mentioned, that we had limitations. So this study was done in Cameroon, where there is no treatment center. So you can imagine that most of these patients were not under hydroxyurea. So that means most of the patient, and it's a rural area, most of the patients were not attending the clinic, not because they don't want to, but because of financial factors. So that means number of VOC might be biased in this, but I think we are happy that SADC has produced a health app that has this data that you can sort of use, so that we control the bias of reporting VOC. I'm not sure whether it has been rolled out, but however, it's an app which will definitely sort of solve this limitation that we had in our study, if anyone wants to replicate the study. And also, the possibility might be the definition of vaso-occlusion crisis might be poor. Some people, they will think that they have VOC even though they didn't have VOC. So I think the terminology as well was another issue in terms of self-reportly in using it in healthcare services as well. So that's what we've listed as the limitation. But then what is our take-home message on the study? I think the study is a very important study for us, because we're able to identify the clinical predictors for patients that are prone or that are at high risk to develop these other organ complications. I've mentioned before that kidney dysfunction also, we saw the G1, G2 genotype for upper L1. So that was very important for us to identify that. And also we can use the number of hospitalization and consultation as a proxy for people that are at high risk, because we know VOC, it's very severe. And these patients that have VOC are sort of prone to like a lot of risk in terms of death. So I think this study contributed a lot in terms of profiling the risk factors or the risk clinical predictors that we can look at it in terms of genetic and clinical information to be able to sort of try to manage our patient. After we've done this study, I think we thought, let's change the gears, because I think in my research group, there is no PhD without functional study. So we did a functional study whereby we wanted to look at the microRNA expression under hydroxyurea. So we collected patients before hydroxyurea for the study, and after hydroxyurea. We wanted to check the gene expression when the patient are using hydroxyurea and when they are not hydroxyurea. Once again, I think our results are sort of preliminary data, because we only had 10 samples for this study to look at, which is the power size calculation. It's not the best. However, the data that we produced for this study, I think it was important data that sort of open our eyes that there's other ways to look in terms of therapeutic targets for our African population that are affected with sickle cell disease. When we did our analysis, we saw that patient that with hydroxyurea and patient without hydroxyurea, you can see the one with red writing where there's an H at the end. It's patient that were under hydroxyurea. You can see the gene expression, but even if you don't know how to read the heat map, you can see the colors. It's different when you compare to the patient that are in black, which are not under hydroxyurea. So you can see there's a difference, and we've identified some novel microRNA. And this microRNA, we wanted to see, because when we're analyzing the data, we realized that there was some patient that lapsed when we were doing the recruitment for 3 months. And we wanted to see if there's any difference with that patient. When we look at that patient comparing with the hydroxyurea, when the sample was under hydroxyurea, and after 3 months it has lapsed, we saw that still we can see the difference. That means the gene expression under hydroxyurea, it's very important, because you can see the expression here, it's very different, and it's the same sample, but then it's the same sample that was collected while the patient was using hydroxyurea, and this patient lapses for 3 months. You can see the effect of 3 months. If you're not using hydroxyurea, the gene expression changed significantly. But when we put these different patients, that when the other one was under hydroxyurea and the other one was not under hydroxyurea, I think the aim here, we wanted to see whatever that we are seeing here, is it not a false positive, but is it a true reflection of the gene expression. We saw that exactly what we saw in the when we compare GS1 without hydroxyurea and with hydroxyurea, it exactly translates to the same thing when we're looking at the different samples that was under hydroxyurea. We're excited to see these novel microRNA, but the other step that we needed to see, what are they targeting, what are they linked to these microRNA? And then we did the network analysis to see what are they targeting, what are they linking. To our surprise, actually, we're happy to see this, actually, to see that all these novel microRNA, they're targeting genes that have been reported to be responsible for the level of fetal hemoglobin under hydroxyurea. This was great to see, and we thought it's nice to find gene, because these genes can be sort of used, or these microRNA that are targeting these genes can be used to explore some of the alternative drug therapeutic, specifically in Africa, because these were not reported anywhere else, but in our studies in sickle cell patient under hydroxyurea. So what is the take-home message from this study? This study, we did, we extracted sample from these patients, and we thought it would be nice to look at functional study, whereby you take the CD34 cell line and you treat those CD34 cell line with hydroxyurea. We did that. Unfortunately, I don't have that data. But then, so we thought it would be nice for someone who's following up on the study to do that, to see if at the control environment, when you do this study, to look at the fetal hemoglobin or the hydroxyurea expression of these microRNA, is it different when someone as is using hydroxyurea and also using penicillin, because we know our patient, they use multiple drugs. But then when you use it in a control environment and you are feeding those cells on hydroxyurea, we were interested to see what is the gene expression, what are the microRNA that will come up, and which gene are they targeting. Is it the same as the one that we have published here? But however, this study, it's a preliminary study, but it also showed that there is a possibility to look at a post-transcriptional therapeutic for sickle cell disease as a target gene therapy, or a therapeutic exploration for our patient. So I think this is the study that I did for my PhD, but I think for a few minutes, I think I want to share what I'm doing now. I think I thought about it so hard, and I thought, oh, maybe I should say from dependency to independency, because the study that I did under the supervision of my peer and my co-supervisors, and through their mentorship, I've managed to move from dependency to independency. Currently, I'm a researcher at UCT, and I'm working on genetics of inherited kidney disease in Africa. So in our first phase, we got a grant from the Female Academic Leadership Fellowship, which is based at VETS, and we got a funding from NRF, whereby we're doing pilot study to recruit 50 families at Barra Hospital. And the second phase, we got a funding from MRC and UCT. In this phase, we are adding 2 more sites to sort of replicate the same thing that we did at Bara. So the aim of my study is basically to look at whole exome sequencing in these patients that are suspected to have a clinical inherited kidney disease, and we are trying to investigate the novel genes or variants that are sort of pathogenic to this different renal disease that we see in our hospital, specifically in Johannesburg. And the second phase will do the same, but in different sites, which is Cape Town and Port Elizabeth. I can't end this presentation without saying thank you to my supervisor, which is in green, Professor Ambrose Wonkam, for the guidance and the mentorship, and my co-supervisor listed below. Dear to my heart, Dr. Gaston Mazandu, may his soul rest in peace. I think he is the one who taught me a lot of analysis in terms of gene expression. And Dr. Gif Pule, my bench supervisor, by then they were called bench supervisor, your co-supervisor. I think now it has changed to be co-supervisor rather than bench supervisor. And thank you to the Division of Human Genetics, Division of Hematology, specifically Dr. Chlodi, who helped me a lot with troubleshooting, and the research group that I was under, which is GenMap. But under GenMap, I was under SADC, which is Sickle Cell Disease Research Group. You can see there's the Sickle Cell Africa Data Coordinating Center, which funded part of my PhD, and my MSc was funded by NRF and UCT. Thank you.
Thank you so much, Dr. Mnika. Thank you for the great presentation and the great work. And congratulations on your work. Actually, it's very nice and interesting. So, guys, please, if you have any questions, currently write down in the Q&A or in the chat, so we can reply for this. For me, Dr. Manika, until the question is written down. So I have a question regarding the status of since you are the expert in the sickle cell, and it's not my field actually, but I'm working in oncology. So what is the status of this in Egypt? Do you have an idea?
Status of what? Of sickle cell disease
Yeah,
generally in Africa. So current
In Egypt, not in Africa.
Oh, in Egypt. In Egypt, we don't have a lot of sickle cell disease in Egypt. I think I've met one of the colleagues from Egypt. They have a lot of beta thalassemia. I'm not sure if you're aware of beta thalassemia. They have
Yeah.
a lot of beta thalassemia, a few of the alpha thalassemia as well. So sickle cell disease is not that prevalent in that region. So that's what we know currently, that there's a lot of beta thalassemia rather than sickle cell disease.
Okay, thank you. So now we have Dr. Yusra is raising her hand. So Dr. Yusra, please start your question.
Thank you so much for the great talk. Actually, I enjoyed it, and I do have a couple of questions. My first question about you are saying that you identified a novel hydroxyurea-induced microRNA that specifically target the hemoglobin, fetal hemoglobin regulatory genes. And my question now for you, that you think that this is could be like a target, like a therapy, we can use it, for like a kind of exploration therapeutic target with these microRNA. Do you think that's like after what happened with SARS-CoV-2 and what we use it with those vaccine, the RNA vaccine? Do you think it's still like feasible, especially we don't have like a driver gene, like a known gene, like the one that target the downstream, targeting genes, the regulatory genes after the fetal the fetal hemoglobin. Do you think just to look like a bunch of biomarkers or microRNAs and a couple of biomarkers will be useful like that, like in one, like a system, not the whole system at one time?
Yeah, I think in a short answer, yes. So you saw that network for the protein network. So you saw that some of the microRNA are targeting BCA11. BCA11 has been well studied and well um, identify as one of the driver for to increase the fetal hemoglobin. So people, they've been thinking a lot of doing like of doing like a therapeutic that is targeting fetal hemoglobin. Even went to like to gene therapy. People wanted to target that, but we thought that it will be great to look at the microRNA that are responsible for the fetal hemoglobin production, mainly specifically the ones that are targeting these genes that have been reported, even they have been reported in Africa as well, that they are responsible for the production of fetal hemoglobin. If those genes, if you look at those genes, we see that patients that have those SNP, they have high level of fetal hemoglobin, and the one that don't have, they have less fetal hemoglobin. We thought microRNA will be a great route actually to look at the microRNA that are differentially expressed in our population and look at that in terms of therapeutic biomarkers.
Okay. Thank you. I do have another question. We know hydroxyurea is like it's a drug. We block cells at the S phase, and it's very strong drug that blocks cells, the division of cells, at the S phase. And you're saying that at the early age you are doing right now some large screening of sickle cell disease for young in just newborn babies, and you think like treatment at early stage will help. Do you think it's critical or to do it at this early stage, and like these kids, they are growing and they have a lot of cell, like they are in development, and will affect others actually cells in addition of the the erythrocytes, the red blood cells.
I think what we did, we let the cells to differentiate. We didn't treat them as they are as CD34. We let them differentiate to a specific level before we treated with hydroxyurea. I think what we're trying to explore, a different stage of differentiation of these cells, how these hydroxyurea respond. Yes, the data is preliminarily, but I think what we wanted to do, we wanted to replicate this in a control environment for us to be able to say this is the right way to follow in terms of looking at the other target of therapeutic, not gene therapy, but other therapeutic management ways, not really gene therapy or anything else.
Okay, cool. Thank you so much. Thank you for the great talk. Thank you.
Okay, do we have other questions? Let's see. Um,
I see Nosipo's hand.
no, I think we don't have other questions. Do you see other questions?
I see Nosipo's hand. I don't know whether
Okay,
it's a historical hand.
so
Nosipo, your hand.
do you have a question?
Hi, I do not have a question. Can someone who raised their hand please talk?
Oh, on your behalf. Okay.
Yes, please. This is like multiple joined under my name, so
Okay. So yeah, I see a lot of nocipos. I raised my hand and it's me. Thank you, Dr. Vitala. Very
Why
interesting.
are you hiding?
No, everyone is called nocipo on the screen, on my screen. I don't know why.
Okay.
Yes, that's why I can see 10 Nocifo.
Yes. So I wanted to ask you, I think Prof. Afani has already asked part of my question, but do you know of any efforts where people are following the biomarkers for hydroxyurea response in terms of drug discovery?
I don't know any research group, according to the literature that I've also read recently, I don't know any research group that are following on that. The only thing that I know the Tanzanian group, they're looking at the NGS panel to look at the variants that are sort of associated with drug metabolism in patients that are using hydroxyurea. However, there is no study that I know that have looked at that in terms of drug development, which will be great to see. However, we couldn't move to that step because we wanted to see if we can replicate the same results in a controlled environment, which we couldn't manage to complete.
Okay. Thank you. Interesting.
I guess it's a project for someone who's interested in sickle cell disease.
Do we have any?
Maybe just a comment. It's a pity to see there's no data about Tunisia, but there's some sickle cell disease in Tunisia too. But the problem, there's a very little research done. But I remember many times we can see through some small projects and presentation from a clinician presenting, some patient with sickle cell disease here in Tunisia. But I can see in your map nothing, no data available. I hope things get better.
Yeah, I think it will get better because when I attended a meeting in Tunisia, there were a few people that were presenting beta thalassemia and alpha thalassemia, and none of them, they were presenting sickle cell disease. And then I did ask a question, you guys don't have sickle cell disease? They say they do have sickle cell, but there's limited research and knowledge around that. So I'm not sure what's happening in Tunisia in terms of sickle cell disease.
Yeah, but there's special actually hospital working just on that at Aziz Othman Hospital, but it's pity, do not see, some research from those clinicians, but it will come. I hope it will come with time.
Yes, definitely. We are working towards that.
Good.
Thank you.
Thank you.
Any other questions? Okay. Thank you so much, Dr. Mnika. Thank you for your effort, and I hope everybody is we're going to attend the next African Pharmacogenetic Network webinar. Inshallah, it will be announced soon. Thank you again, and see you soon. Thanks, Dr. Mnika. Thanks for the attendees.
Thank you so much for having me.
Bye.
Thank you.
Bye. Thank you.
Thank you so much. Thank you, colleague. Bye.